Okay, some thought today, why microbiology in NASA? Everybody think it is an engineering institution. What the hell we are doing there, right? But think about it. The scientists are the one who drive the whole institute, whole agency. You have to ask the right thing. In here, we are asking, are we alone? Did we come from somewhere? Did we get implanted here? So if there is a case for one case is Mars, we might have exchanged the meteorite and then started to 3.7 billion years back. That molecule get adapted to the conditions of the earth and then flourished from the primitive single cell bacteria to human we are. Okay, so we can ask that question. To do that, that is the science. The biology is the main thing here that drive the 99% of the engineering course, engineering fact. Obviously, the one that we landed in Mars is the engineering fact of the century. 20th century is engineering feat of the 20th century. Astrobiology, I already said, if you are sending a mission to look life in other planet, that is first you do the robotic mission. So robotic mission, if you're looking for life better, you clean yourself. Otherwise, you do not know what you are measuring. That one I said the planet and planetary protection. That is also robotic mission. Then you look for life and you should not contaminate now. If you contaminate, whether the red planet which you are seeing right now in Mars may become green otherwise tomorrow, if you are not taking care of it. Same thing here also, when you go to the national park and other things, take your own garbage along with you, right? Same thing that we should not contaminate our planet. Now, after these robotic missions, we are trying to go, the human are trying to go there. So for that, how the microbe differ, how that changes, whether there is positive or negative influence towards the human. Here, that's what the human research program, that whether it affects the crew health. The other one is you can not take everything with you if you are going to go and explore other planet. If you want to have a sustainable life in Mars and other planet, better take the best out of the microbes with you, even making oxygen, removing carbon dioxide, anything that making food. Okay, so everything that you think of, for a sustainable life, you need microbes. So while doing that, we found several novel microbes that are, producing something, and that I will touch something. And then unusual microbes characteristics will be helpful. For example, if it forms biofilm, how you are going to remove it. And the whole genome sequencing will help us to understand those characteristics. And then proteomes, metabolomes, metagenome to phenome, the things that you do not know how to culture. You said emerging disease, emerging pathogen, right? So that one, we can come back from the metagenome, from the genome-based analysis to culture them. Once you have in culture, then you can develop. Okay, next one is space station. This is what I'm my sampling coming through. Anybody knows this guy? He's Scott Kelly. He's the one who spent in space station for more than a year. Almost a year. This is the guy, Captain Terry Wirth. He's the one who collected our samples. And I had about 9 years of sample collection from space station. Astronauts can live maximum 6 months. Some people extraordinary like him are living 1 year, but maximum is 6 months. So every 6 months you have the crew changes. So in 9 years, I have multiple crews. So that means what? The collection people, either they are efficient sometime, they are not efficient sometime. They may shed their microbiome. All these things are also varied. So we have a baseline study also. This one is a video, but I'm not going to show all the 9 minutes of this video, but some of the things I'm telling here, we monitor how they collect the samples. Okay, so here he put the samples to collect the air samples. This is air sampler and then you put an appropriate filter there, and then collect. So he comes and put it a kind of air filter. It's not a petri dish, but it's the same size. He put it. Look at it. He's not wearing the glove. So we are monitoring, and then telling him, "Hey, you are doing this not correct. Let's wear the glove." And then, how difficult wearing the glove? You guys are very good in when you are doing the surgery and other things. You are changing your glove, but still here, but your mouth and other things are covered, and you should not touch, right? So somebody is helping here. It's not happening there. He's struggling to have the sterile glove, that is different from normal glove, and it is floating in the because of the lack of the gravity. Okay, so he has to struggle in putting those things. So that kind of thing. So after that, he get the samples, wipe the samples appropriately. Now he done the gloves, and then he's taking the wipe, because he's touching the wipe, because we don't have a better sampling collection device that collects 1 meter square efficiently. So he's collecting these kind of things that we are giving it to him. See, a lot of things these are Velcro, sitting on his various part of his body, and that's how we are collecting. Still, it is not a sophisticated sample collection, but we are collecting some samples that will be coming back. While doing so, what happened? Sometimes the following thing happened. Somebody swing by you. That means when you are doing a surgery, if somebody is not clean, that person will contaminate. So that's what is happening here, and we are collecting. Well, we are collecting the people who are inhabiting in the environment. Sometimes, it is not only for ladies, but anybody will chatter things next to you. If they want to say something to you, they'll come and then they do some experiment next to you. Okay, they are non-stop. They are not wearing the there is no corona there, so they don't wear mask there, but they speak. So his oral microbiome, her microbiome will also be there. So all those things are there. I'm coming to this, because I have to go faster here. This is the one I'm going to talk today. First of all, as I said, challenges, biofilm, how you can develop certain, antimicrobial coating. That helps. Otherwise, biodeterioration. 10 years back, you might have built a house. Now you are seeing some problems there. You can easily change, but that's not going to happen in space station. Space station is 22 years. Either you abandon them, destroy them, which was more than $200 billion worth of stuff there, or come up with a technology to develop something new, different. And the other one I also told, because you guys are a medical professional, you need to know the problems and the associated things. So in space station, microbes develop more antimicrobial property, more virulence property. So you need to come up with some sort of antimicrobial thing. And also it produces some virulence factors, right? So that resistance is the biggest thing. So I will tell the Kalam appears on his story. As you know, this is named after Abdul Kalam, a genus, okay? And then we also need appropriate instrumentation to do, you are sending your samples for sequencing. You don't have a FedEx in space station to bring it down and then do things. You have to do everything there. How many moving parts are there? How do you collect even the transfer the fluids from one to the other, right? So those are the things are there that we need to work on. So I'm not going to cover all these things that I worked there around 25 to 27 years now. This is Elon Musk standing next to his SpaceX. So that is the one is transporting things to space station. So that means it is there is one source, right? And the other one is, this is Earth analog. Crew time is very hard to come by. 1 hour of crew cost about $2 million. Astronauts are not getting the money, but you are sending things, all other things. If you calculate things, each experiment that I'm sending it for space station, it should be simple. It should not consist of astronauts' time. At a given time, 160 experiments are going side by side. Astronauts, only 6 people. They may not be microbiologists. They may be engineers. So they can do switch on, switch off. That's it. That is his microbiology experience. That means you have to bring such a kind of simple instrumentation to space station. Okay. Anyway, so first you need to know what is the baseline there. That's why the inside the space station, and the other one is things in space. What is happening? All other things are, I'm not going to talk today, but anybody interested, please talk to me through Dr. Damodaran, and we can do that also. So these two I will try to cover. We came long way. These are the nothing but a simple petri dish, grow them. All of you might have done the microbiology, to the sophisticated metagenome as we know today for ultra clean biomass environment. Whatever we are doing is drawing the blood out of the rock. That means very, very, very low level, below detection limit actually. Okay. So what we do, we collect samples as I told you, I showed you, okay? And then we also don't forget the traditional microbiology. Without that, you cannot develop countermeasures at all. Without that, you cannot come up with some sort of requirements to place. Everybody say, I'm having a countermeasure development already. I'm killing the bacteria, leaving behind the DNA. I don't care, right? That is not going to do anything. But if you have the microbes, whether you are leaving behind or not, you will know, right? And also we develop technology how to selectively remove the DNA and other things as a remnant, only look for viable microbes. For all these things, you have to, the other one is biofilm, right? You have a spacesuit, but you don't have so much to pick. Astronauts come 6 months after that, he or she cannot have her own stuff. It is there, S, L, M kind of thing that you pick your size and then use it. What that mean? You are leaving behind something and the other person is wearing it, right? You don't have a laundry there. You have some chemicals to destroy it for sure, but it is not 100% sure. This is the texture is the spacecraft spacesuit material, okay? And then this is the biofilm forming, the red one or the rhodotural mucilagiноsa that actually, making this biofilm on the spacesuit. But as I said, this is rhodotural mucilagiноsa. This is having two different kinds. One is clean, round. The other one is spiky stuff. Those spiky stuff are the one producing this thing. So not all the things that some subsample is doing. We need to develop some countermeasure. Here is the one that actually kills this kind of bacteria, and that you coat on the surface. This is stainless steel. Medical institutions also work with the different materials to look for it, right? So this is a water processor materials. 5 materials we tested all killed the Pseudomonas aeruginosa, no problem. But rhodotural mucilagiноsa, that is not get killed. Why I'm saying is not one size fit all. We have to have multiple things. In this case, this is what is happening. If your surface coated with something and bacteria come in contact with that, yes, it get killed, including yeast or something. But when you have the detritus material there, the other one can sit on top of it. Now it is not in contact with the material, right? So that is what is happening. This is bigger, this is not that bigger. So here get killed, but here it is not. Okay, this is for stainless steel. This is for Teflon. You have medical devices with Teflon as well, right? So it is okay and get killed, all the Pseudomonas, but not rhodoturula. This is, this is inconel, this is titanium, and mainly because of this thing. So this gets settled, and that is where the things get killed, but the other things are sitting on top of it. That's why there we started seeing so many novel bacteria. This is the one that I worked with the MTCC people. Miles Raj is my collaborator there, and then Solibacillus calamii. This you are not when you are looking for all these things, you are not going to name each and every bacteria. This has to do something. It should have some characteristics. It should be problematic or it should be helpful. Okay, so Solibacillus calamii is, as you know, this is after my virtual mentor. Everybody's mentor he is, and this producing exosporium. Some of you may be working on Bacillus anthracis, so on Sirius. So that has this additional layer. This is called exosporium. You can strip that off, but still it will be alive. But that help to get protected from radiation. This is made up of glycoprotein, and that can be used as a sunscreen. So that can be developed for astronauts. Spacesuit get coated, astronaut visors get coated, and also it is biotechnologically helpful for the Mother Earth as a sunscreen development. Okay. Then another thing, Methylobacterium ajmaliae. Damu knows. He is my senior, Dr. Ajmal Khan, and I named after him. And why I am characterizing this. This is a rod-shaped gram-negative bacterium. It helps to produce a hormone that promotes the plant growth. As I said, you cannot take your tons of soil with you to grow something. So you have to use moon soil or Mars soil to see whether anything growing. For that, you feed this hormonal stuff. This may be helpful for the soil to get fertile. You may be asking me, hey, first go and make Sahara Desert to green. Yeah, we are also testing that. Yeah, whether it is going to be useful for our Mother Earth or not. So we are using that too. So I named this after my friend, and he's not my immediate senior. I mean, he's my immediate senior. It's not. He's fish microbiologist, marine microbiologist. I am marine. See, he's marine biologist. I am marine microbiologist. We communicate and talk more thing, but he's as simple as Abdul Kalam. Very, very simplistic man. If you are simple, you cannot come up into your administration, but he's a very good scientist. That's why I named after him. The other one is chloramphenicol. So most of you prescribe chloramphenicol as a generic drug without knowing what bacteria it is. Some bacteria is actually growing up with that. Okay, I'm saying growing. It is not withstanding. It is growing. We put it in the presence of the things at appropriate microgram level, that we use it to contain bacterial growth when you're looking for fungal growth, right? So all of a sudden, some of the time you are getting this actually also producing biofilm. And I named after Kate Rubins. She is the astronaut who sampled my things from space station, and also she's the first human who sequenced DNA in space. She's a molecular biologist from Stanford as well. Okay, so now then it is not only bacteria around, now fungi. Fungi is also producing biofilm. So these are the forms of these cotton-like structures that you are seeing. What you are seeing in the middle are those conidial spore. Okay, so these things are these are hyphae, fungal hyphae, and this was novel, isolated from Mars 2020 assembly facility. I named after one of my great colleagues. He's the one who collected all the Chernobyl fungi. He's a mycobiologist. So Thomas Torok, so I named after him, Torokii. And then another example for yeast. So you saw bacteria, you saw a fungi, you are seeing a yeast. Anybody working on Cryptococcus here, neoformans? So you medical professionals may know Cryptococcus neoformans is a deadly yeast infection. One of the characteristics is nothing but the Titan-like cells. It forms, balloons it when it gets into your system. Okay, so this is the first novel eukaryote isolated from space station. I named after a person, philanthropist, who spent money to do the research to Chris Mason, actually, Damu knows, and his name is Igor Tulchinsky. He spent $5 million for this, so for the space station sequencing project. So we named after him. Here is the thing, what I said. Here is a normal yeast cell which all of you who are working on yeast may know, under 1G. 1G means normal gravity, okay? And if you are growing it into the 5% CO2, which is the human thing that you can mimic to some extent, it is still the same, a little bit bigger, but it's still the same. But when you grow the double punch, microgravity plus CO2, it balloons. This is the kind of things happens for Cryptococcus neoformans when it become pathogen. We are still working on this. Multi-omics analysis is being carried out right now, and we are still working on this, okay? So one other thing that is very interesting is, yeah, these are all there. It's not a rare event. People may ask, it may be there because it can happen in your home. It can happen in your hospital. You know, why do you need to worry? Persistent is the biggest issue, right? So let's take the BSL-2, all of them. So now you are not able to see everything, but some of the one that is bigger font are related to the incidence of it. That means it is there in numbers. That's one. Is it one time? That's another thing, right? So you can say, it is one time, I'm cleaning, it is gone. If it persists, that is what is happening here. These are flight 1 to 3. Flight 1 to flight 2, 3 months difference. Flight 2 and flight 3, we planned 3 months difference, but SpaceX 6 blown away. NASA had no vehicle to go there, so 1 year. So flight 1 to flight 2 is, 3 months, and flight 2 to flight 3, 1 year. So that means it is still persisting. And third year, actually, it actually spread to other locations. These are the locations of the space station. Exact same locations. We went there to collect the samples that I showed you before. The astronauts collected. And then the other one is that initial 2 different strains same or different? For some reason, this here, I'm not seeing anything. Other Staphylococcus aureus popped up rather than Klebsiella pneumoniae. As you guys know, it is a pulmonary infection, Klebsiella pneumoniae. If you have a corona plus Klebsiella, it is a big problem, right? So is the spread of this species, which is an opportunistic pathogen coming from one strain. Klebsiella pneumoniae, you guys know it is an opportunistic pathogen, it is well treated, but can you treat in an immunocompromised person? That's a big deal. Astronauts are very, very, very healthy, but when they be there for 6 months, psychologically disturbed, physiologically compromised, because they don't know where the head, where the toe is. They have the fluid exchanges from top to head. Right now we have a different. How many of us can stand upside down even 5 minutes? Right? So they are there for 6 months. So they are immunocompromised. A benign microbe, even a simple E. coli, may be a problem. So we need to come up, how do you find? We have the sophisticated technology like metagenome analysis to be carried out. And then we have the data in a public database to mine. And is this by one single strain? If so, how do you eradicate? Gene therapy, like phage therapy. If you come up with the appropriate phage that actually put that, then now you can mix that with that. So now for that, you need a basic science to understand. So one of the things that is metabolic modeling, how this Klebsiella is helping, or how do we kill it? Now let's see how it is helping to produce some antifungal compound. So you can do the metabolic modeling of all these things, and then this Klebsiella is acting upon. This is parasitism. That means if you have this mark, this is parasitism. This is mutualism. Commensalism means mutualism. Okay, so here the rhodotorula are actually killing Klebsiella. Rhodotorula are killing Klebsiella, but Klebsiella is killing Aspergillus penicillium. You can predict this. Is that right? You have to validate it, because we have skepticals sitting in front of us saying, I am experienced, you tell me how you can prove this, right? Okay, this is the fungi, Aspergillus fumigatus. All of you know BSL-2, pulmonary infection. Again, it is an airborne thing, okay? This is hyphae, And these are the conidial spores that is very healthy under the regular conditions. For the past couple of decades, microbial monitoring is happening based on omics technology.
Omics includes everything inside the microorganism, not just their species identification through their DNA, but also what enzymes they're producing, what proteins they're producing, and what metabolic they're doing. And as we study microbes in space, these are key things to take into account because those can give us clues and hints as to how microbes are changing and adapting to the space environment.
So currently on the International Space Station, there's no mechanism to be able to process samples. The process for microbial monitoring on the International Space Station is for astronauts to collect samples from either surfaces or the air with swabs. Once the samples are ready to be returned to Earth, it gets put into a capsule and then it lands into the Pacific Ocean. A boat then comes to collect the samples, which is then transferred back to different NASA centers. So it's very important if we could cut all of these time and these losses by performing sample processing on the International Space Station itself.
So one of the things is we need an automated sample processing instrument, which we call them as SPI system, that will be helpful to measure the microbial population in space in a closed system during the long travel.
The SPI is a microgravity optimized DNA extraction device that's ideal for use on the ISS. The crew time will only spend about 1 to 2 minutes per sample, and then they could use our device to process multiple samples simultaneously, which means the data that they collect eventually will be more reliable, simply because more sample and more repeats can be performed with our device.
So once an astronaut collects the samples, it would take only a day for us to get results back rather than about 4 months, which is now the current protocol.
But we do not know whether it is going to work in space because fluids behave differently under microgravity.
The SPI device can successfully extract DNA on the Earth's surface. However, the fluid properties of liquids in microgravity environments require novel engineering approaches. Specifically, a new well-shaped design will be required for the SPI to function in microgravity.
So if you don't design for microgravity, you can have fluids in all sorts of places. You can have the liquid on the top, you can have it on the side, you can have it on the bottom, you can have droplets ejecting, you can have geysers created, you can have all sorts of things where you really just don't understand what your fluid's doing or where it's going, and it can be very dynamic. And in this project, what we're doing is we are taking a closer look at the well plate design, and we're modifying that so that it works predictably and efficiently in a microgravity environment. In a system that's designed for microgravity, you'll see the fluid at the base of the container, and it'll be just like you're in 1G. It'll seem like gravity is actually present because all of it will go to this well at the bottom. And so you create a predictable, simple system that's passive and is self-correcting.
JPL and collaborators developed a pipeline, a microgravity independent pipeline, starting at the species, extracting their DNA, prepping the DNA, make it ready for the sequencing step, sequencing it on the biochemical sequencer, the Oxford Nanopore Minion, and actually be able to analyze the data on site, on the space station.
The data that we receive from the MinION can then be sent to various bioinformatics pipelines.
We take those files and process it as per the codes and open source software that are available to us. We do a quality check analysis. Each and every sample is checked whether we got the right results. We check it through the rare fraction charts, and the rare fraction chart tells you what type of assembly you will get and what type of organism or diversity you will get out of it. Once we have the quality reads, with us, we do the identification and function analysis. The function analysis tells us what the system itself is doing here, which tells us what type of organisms are there in the sample or what type of processes they are doing. We don't want anything that is harmful to humans.
Now that we have a pipeline put in place where we have DNA extraction on the SPI, sequencing on the MinION, and bioinformatic analysis, we had to put this pipeline to test. We decided to test this in a variety of environments that fluctuate from low biomass here at JPL at a clean room, and high biomass in Yellowstone National Park on multiple biofilms that form in hot springs.
The hot springs of Yellowstone National Park provide an ideal environment for studying microbial ecology because they're remote, they have high biomass, and they are extreme environments. And this is analogous to how the SPI device will work on the ISS. It's a remote environment as well.
And what's so great
are my group people, either postdocs or scientists there working. So when you are developing such kind of technology, think about you are giving your blood samples to for your regular checkup. The doctor will say, come later 3 days, then I'll do it. You will get the results, even in the earth now, right? Think a system there right now next to the doctor's office, then he can prescribe the medicine. Until then, he will give you a generic medicine to you. But if he comes to know that something is really a specific thing, then he can tune his treatment differently, right? So likewise, and also similarly, we wanted to firstly extract the DNA. To extract the DNA, to get this machine, it has to be miniaturized, right? And it has been tested in the clean room, as you have seen, and it has been tested in the Yellowstone. It has been tested in the back of the vehicle, which is a vibrant thing, 85 miles per hour, which is 140 kilometers per hour. You can do it in India, okay? But in US, so you run on the back of the car and then see how it is working with the vibration. Everything worked fine. So we published this paper, but important thing is microgravity. Is it going to work? Because you saw what the problem in the microgravity. Fluids will fly everywhere. One of the astronauts who was from Italy could not bear drinking coffee in the Tetra Paks. He wants an open cup. So he threw this idea, I want an open cup. So some material scientist people designed that coffee cup that you have seen. So astronauts can sip the coffee in an open cup now, because even upside down it is not going to spill because of the property of the material. So same materials we are using it. It's different design, but same material we are using it for our purpose. Okay? Now, in the ground, you have to test the microgravity. There is nothing you can simulate microgravity here, other than this parabolic flight. What is it parabolic flight does? 20 seconds of weightlessness you can get. So if you have 1.5 or 2 hours flight, every 20 seconds you get 20 seconds, and go up, and then come 20 seconds, so 40 times you will get, so you will be able to get 2 to 3 minutes of your time. So every single time you can test one and test second. This is important before you fly. What I am trying to tell you, these are the challenges we have. It is not that I found something, some electrochemical properties done here on the earth. Is that fluidics that you have can be measured appropriately, can be contained appropriately. Otherwise, you are going to contaminate the station. So this is my postdoc, Seth Parker. And then, if you take the current microbial monitoring system, you collect this swabs or samples and store it, and then you saw that how it is being dumped into the water. And then all these things take around 4 to 6 months to measure what it is. If you have a DNA extraction system that is working under space, all these things can happen within few hours. The time taken here will be roughly 9 hours to get the same thing. But first let us test whether it works under parabolic conditions. So around 6,000 meters, the NASA flight, NASA aircraft fly, and then up to 8,500 meters it goes, within few seconds, 20 seconds, you will get a hypergravity. Then you will get weightlessness, 20 seconds, like that. Okay? So under 20 seconds, this is a real picture. My postdoc's picture is there. And then this will give you an idea what will happen under the microgravity. Most of you know that pipetting, and then all the pipettes are flying everywhere if you are not containing So fluid transfer has to happen in a contained form, otherwise this is the one you are going to happen in space. Okay? So we took it into this 0G, and this is the instrument that is getting into there, and then we are loading the cartridges. This is called microtitan, a sample processing instrument. We load it. All those things are contained in the surface of the 0G flight. Okay, and then this is the flight pattern. What are they doing? Simply jumping and down? No. This is 20 seconds. They are experiencing that weightlessness. This is called vomit comet. Everybody vomit. All this flight is every time after that they clean thoroughly. Otherwise you will get only vomit things around. But anyway, so you see those spikes, that is when you get the weightlessness. It is only 20 seconds. With that time you have to do your experiment. It's not only your experiments going on. You have 50 different experiments going on at the same time. Okay, this is the successful thing. So he gets strapped on the floor. Okay, that guy, you see that guy gets strapped, and then soon you will get this weightlessness. So now they get weightlessness, and he is observing what is happening in that instrument. The guy on the left side is a very experienced. He is swimming nicely. All of them are falling down, upside down. They are first time riders.
Yes! Yes!
And then he said it is nicely moved from one well to the other well. Okay, that you have already seen. Instead of pipetting, automatically the things are moving from one well to the other well. Okay? So with this, in the interest of time, I'm not going to go in details about several things. This is not one-man show. This is the combination of several people over the period of time, 25 years worth of my thing, which I have shown only 2 to 3% of my research here. But this was possible because of these people.